Have you ever had a professor explain the research to you and you thought it sounded really cool, but you didn't really understand what it meant? So have I. And that can make getting into undergrad research really, really hard. Hey guys, my name is Leen and welcome to In Plain Science, where I'll be demystifying undergrad research on campus. And today we're going to be taking a look at Dr. Lessner's lab. Dr. Lessner's lab studies methanogens, tiny microbes invisible to the naked eye, that produce methane as a byproduct of their metabolism. Methanogens contain an enzyme called nitrogenase, a specialized protein that speeds up the conversion of nitrogen gas to ammonia. This process, known as nitrogen fixation, makes nitrogen available to other organisms. Nitrogen is critical for plant growth and survival, yet they can't use it directly from the air. Today, we rely on synthetic fertilizers, which often pollute waterways, but methanogens are a promising sustainable solution.
What we hope is understanding fundamentally how this enzyme works in methanogens, that could lead to other scientists being able to take this information and engineer plants to actually fix their own nitrogen from the atmosphere.
Oxygen is toxic to methanogens, so preparation and handling of the media has to be done in oxygen-free chambers.
I'm making the growth medium that we use to grow the methanogen. So this will turn from a dark blue through to a dark pink, and then eventually clear after it's been heated up to sterilize it.
The most important thing that you learn early on about these microbes is they grow very slowly compared to more typical lab organisms like E. coli. So a growth study is going to take several days at least.
This is how we check the growth of our samples. So we look for the optical density. Media goes from this just completely clear color here, and then you can see just the large number of cells. You can use the spectrophotometer to check out the optical density. So it tells you how much of that light gets absorbed by all these cells. The more cells, obviously, the more growth.
So I was looking for a lab to do my research in, and I originally looked at Dr. Lessner's work and found it really interesting. I didn't know that much in the beginning, and then once I met him, I was pretty sure he's just a super kind and great mentor, and I have enjoyed being in the lab ever since. I suggest giving yourself the time to really find the lab that works for you. It's nice to be able to talk to multiple professors and get to know more about the research.
So overall, Dr. Lessner's lab is really trying to utilize the potential of methanogens to create sustainable solutions. Thank you guys so much for watching this episode of In Plain Science, and I will see you next time.